Okay, so to talk about photosynthesis, we need to talk about light. Um, and we'll talk about specifically visible light. We're not going to talk about any of the other forms of light. We're just going to talk about visible light, so the Roy G. Biv. Something key to understand about light is the longer the wavelength, the lower the energy. Conversely, the shorter the wavelength, the higher the energy. So as we get that crest and trough, as that frequency increases, we're going to have a higher energy electron available. Just definitions here. Wavelength being the distance between the crests, and the length of the wave determines the type of energy that we're talking about. So basic definitions. Go ahead and read through those. Know that we're talking about electromagnetic spectrum, that light can also be referred to as electromagnetic energy or electromagnetic radiation. Sorry guys. And that visible light has wavelengths, so the different colors we see produce different wavelengths, which means they're absorbed differently. Light behaves in a way as if it has very discrete particles, and we call those particles photons. P-H-O-T-O-N-S, photons. Okay, so we're going to have a much shorter wavelength down here. Our peak and our trough are going to be closer together. On the longer wavelength end, they're going to be more in the middle. Okay, so there's just a practice question for you to go back and look at. If I'm looking at this, or if I'm trying to do, trough and peak, so let's just do, I'll try and do this. I don't have a purple, but I'm just going to grab a blue here. Okay, and I'm just going to do this, and we'll do, okay, so this right here to right here, those are both of the crests, and the wavelength is the distance between crest and crest. If we look down here, here's a trough, and here's the next trough. Okay, so if you're looking at crests versus troughs or determining wavelength, that's how you do that. Okay, sorry. Okay, so pigments. Pigments are what's going to absorb the visible light. So it's going to absorb a specific wavelength depending upon the color it is. Something that's red is going to absorb the part of the visible light that is the red wavelength. Something that is green will absorb the wavelengths that are associated with the green portion of visible light. Okay, any of the, holy cow, you guys, any of the wavelengths that aren't absorbed are reflected or transmitted, and they're bounced off. Okay, so the reason a leaf appears green is because the chlorophyll reflects and transmits that green light. So something that's red is going to absorb all the colors except the red. It's going to bounce off the red, you're going to see the red. Anything that's green is going to absorb everything else, bounce off the green, you're going to see green. And you can do that for any of the colors, or any variation of the color, light blue versus dark blue, medium blue versus darker medium blue. Okay, so it's all about which wavelength is being absorbed, and what's being absorbed is what is everything but what you see. So what you see is what's reflected. Okay. Spectrophotometers measure that. The primary version, or the primary pigment for photosynthesis, is chlorophyll, and chlorophyll comes in a bunch of different varieties. Chlorophyll A specifically, sorry if you heard part of that sneeze. Chlorophyll A specifically is the one that works predominantly for photosynthesis. And if we look at how it absorbs light and what's reflected off of it, we know that the blue-violet or the violet-blue and the red light work best for photosynthesis. For chlorophyll A, it's gonna absorb those wavelengths the best. It's the main photosynthetic pigment, like I said, but like I said, it just isn't chlorophyll A. There's also chlorophyll B, and chlorophyll B will absorb pigments that help kind of broaden that out, so you don't have to have that one tight little wavelength that is the only one that will work for photosynthesis. There are also other pigments involved. So what they do is they absorb other lights that would be damaging to the chlorophyll. So the idea is to try and protect the chlorophyll whenever possible. Okay. This talks about the electrons and them going from a base stable state into an excited state. So as we absorb this photon of light into the chlorophyll, holy cow, excuse me, into the chlorophyll molecule, the electrons are going to be pushed up. We're in an excited state, we're unstable. As they start to drop back down to the base or the ground state, we're going to give off a little bit of heat and we're going to give off a little bit of fluorescence. Okay, 2 whiteboard questions for you. With answers. Okay, so what's a photosystem? So, hang on. Okay, so we need to talk about where photosynthesis happens and how it happens. All right, so the first thing we have to understand is a photosystem. And a photosystem looks something like this. This is a generic photosystem, okay? So it consists of a reaction center, which is a protein, okay? So I have a reaction center, which is a protein, that's right here. And it's surrounded by these light harvesting complexes. That's the purple, the pinky purple, this is the pale lavender, this is the other purple around it. Okay, so where are we? We're in the thylakoid membrane. Remember, the thylakoids are stacked up as grana. So I have this interior, that hollow space in the middle, right? And this is the liquid outside, this is the stroma. Okay, so the photon of light is going to come down, and we're going to have that photon of light hit these light harvesting complexes. And inside the light harvesting complexes we have our pigments. We have our pigment molecules they're bound to this protein. Okay, so what's going to happen is we're going to harvest this, we're going to get this energy, we're going to transfer electrons to. We're going to send this energy one to the other, and here in the center, in our reaction center complex, we're going to find a very specific special pair of chlorophyll A molecules. What's going to happen when they receive that energy is they're going to excite those electrons up to the reaction center, up here to the top, to the primary electron acceptor. Okay, this primary electron acceptor in the reaction center gets excited because we've got this energy coming up from the electrons. Okay, and we've got this transfer of electricity. Okay, so that's the light harvesting. That's how we're going to harvest the energy from the light. We've got the photon coming in, and what we're going to do is we're going to move these electrons. We're going to excite these electrons, and you're like, well, wait, where did the first pair of electrons come from? I'm going to come to that in just a second. I'm going to back up a couple of slides just so you can see. If you go through your notes, you have all of these words. They're all there. So if you didn't get them written down, they're there. Okay. All right. So this is a generic photosystem, and that's what's going to happen. We'll get to the specifics in a second. But there are actually 2 versions of this photosystem. There's a photosystem 1 and a photosystem 2. And in actuality, the one that functions first in photosynthesis is photosystem 2. And you're probably thinking, well, why is it named photosystem 2 if it acts first? Well, it's because they named them in the order that they discovered them. So they discovered photosystem 1 first, not knowing what it was, they named it photosystem 1. Then later they got more of the picture and they discovered photosystem 2, and they discovered that photosystem 2 actually plays its role in photosynthesis before photosystem 1. So that's why the names are reversed and backwards and messed up. But basically what's going to happen is photosystem 2, or PS2, is going to react first. And what it does, it's best at absorbing light that's a wavelength of 680 nanometers. Okay. Photosystem 2 absorbs light at a wavelength of 700 nanometers. Okay. Chlorophyll A in a photosystem 2 gets excited with 680. If you're chlorophyll A in a photosystem 1, you want wavelength 700. So remember I said these were different. I could have one that looks like this and it's a photosystem 2, or I have one that looks like this and it's a photosystem 1. But if this is a photosystem 2, then when we get the energy from wavelength 680 here, that's when we're going to see these electrons shoot up to the primary electron acceptor. If I'm a photosystem 1, it requires the wavelength of 700 to make that happen. Okay, so what then is going to happen is we're going to move those electrons, right? So we're gonna basically have an electron transport, and we can do this in a straight line, in a linear fashion, or in a cyclic fashion. So these are your two compare and contrast. I'm going to skip over them for a moment because I need us to look at what I'm actually talking about in part of that, because we talk about linear and electron flow. So this is your summary slide to start, and then we're going to talk about it. So there are two possible routes for those electrons. We can either go through linear flow or we can go through cyclic flow. Linear flow is the most common pathway. It's the primary pathway. Both photosystems are involved, so we're going to use PS2 and PS1. And what we're going to do is we're going to produce ATP and we're going to produce NADPH. NADPH is an electron uber. It's just like NADH, except it has a P designation in it for photosystem, or if you prefer, think of it as plant. But what we're doing is we're going to shuttle around electrons. The linear flow looks like this. So here we are. This is photosystem 1. This is photosystem 2. We're going to absorb this light. And I promised I'd tell you where we got that first pair of electrons. We're going to get them right here off of water. So we're actually going to split a water molecule. We're going to release the oxygen out, and we're going to use those electrons in the hydrogens, and that's what's going to be right here at PS. So we're going to absorb the light. We're going to transfer this energy back and forth, back and forth. We're going to excite these guys at P680, at wavelength 680. We're going to shoot these electrons up to the primary acceptor, and then we're going to tumble them down an electron transport chain, step by cytochrome, step by step by step, okay, until we land here at photosystem 1 and these guys. Okay, in the process of this electron transport chain, we're going to generate ATP. Not a lot of it, just a little bit. Then what will happen here is we'll excite these electrons. We're in photosystem 1. They'll go up to the primary acceptor. We're going to do another little mini electron transport chain. This time we're not going to make ATP, but what we're going to make is NADPH. And this NADPH, as you already know, is an electron uber. It's going to carry around electrons, and it's basically a little energy packet of electrons. Okay? If that's not clear, this is the other thing the book does, is it creates it like this. So this photon excites these electrons. They come up to the primary acceptor. They tumble down this mill, or this electron transport chain, where they fire another photon, where in combination with another photon, we're going to excite electrons. We're going to scoot them up, and then we're going to tumble them down, and we're going to make NADPH. All right, that was the linear flow. So this coming up is the cyclic flow. And the cyclic flow is different. It only uses photosystem 1. Okay? It only uses the second half of that process. It produces ATP, but it never finishes out to produce the NADPH. So the only thing we get from cyclic flow is ATP, which is really satisfying. I mean, that's an important thing if you need ATP. So what we're going to get is we've got this cytochrome complex. We're going to run through all of these guys. We're going to primary push up in photosystem 1. We're going to excite those electrons with a photon of light. They're going to go up, excite this primary acceptor, and instead of, so in linear they would have tumbled over here and made NADPH, they're not. They're going to cycle back into this cytochrome complex, into this electron transport chain, and we're just going to sit here and spin in a loop. As long as we have the wavelength of light coming in, we're going to sit here and spin in a loop, and we're going to produce ATP. Okay. Some organisms, like the purple sulfa bacteria, they have a PS1, but they don't have a PS2. So their only option is to do cyclic flow. They don't even have the opportunity to do primary flow, or linear flow, sorry. So that We think that electron flow, the cyclic version, evolved before before linear flow. We don't actually know, but that's what we believe just based on the organisms that have it. And the other thing about cyclic electron flow is it helps to protect cells from light-induced damage, because you can run photosystem 1 with a different wavelength with limited light, and it can actually be protective to cells under certain situations. Okay, so questions for you? Answers for you? More question, or rather another question. Okay, I'm going to pause here and we'll pick up with Calvin's cycle.